Hi everyone, this is Adil. Today I'm going to talk about structure of hair. Hair is one of the appendage of a skin. There are various appendages of skin which include hairs, nails, sweat glands and sebaceous glands. Today I'm mainly focusing on the structure of hair follicle. Let's see the structure of hair follicle. The tip of the hair follicle is known as ostium, and this portion is known as the shaft of the hair follicle, and below the shaft there is the root of the hair follicle, and below the root of the hair follicle there is a bulb of a hair follicle. Within this bulb there is a inverted V-shaped structure or V-shaped bulge which is known as Adamson's fringe. Adamson's fringe is a inverted V-shaped bulge which is present within the bulb of a hair follicle. The root of the hair follicle is associated with sebaceous gland. Here is the sebaceous gland, and this sebaceous gland is having duct, and this duct is having opening of sebaceous duct. These hair follicles are attached to erector pili muscles. Here is the erector pili muscle. The hair follicle can be divided into main four parts. Let's talk about those four parts. The portion which is present between the ostium and sebaceous duct opening is known as infundibulum. The portion between sebaceous duct opening and erector pili attachment is known as isthmus. The portion between erector pili attachment and Adamson's fringe is known as stem, and the portion between Adamson's fringe and the base of hair follicle is known as bulb. Infundibulum and isthmus forms the upper hair follicle, and the stem and bulb forms the lower hair follicle. Just remember a very important point here, that based on attachment of erector pili muscle, the hair follicle is divided into upper hair follicle and lower hair follicle. Let's see this question. This question has been asked in DNB 2017, that Adamson's fringe is a component of. It is a component of bulb of hair follicle. As you can see here in this picture, this inverted V is a bulged portion in the bulb of hair follicle, and it's the inverted V, and this inverted V structure is known as Adamson's fringe. This whole area is the bulb of the hair follicle. Adamson was a scientist who noticed while studying cases of tinea capitis that the fungal hyphae and fungal spores, here are fungal hyphae, these are fungal spores. He noticed that these fungal hyphae and fungal spores never intrude or never invade the Adamson's fringe, because Adamson's fringe is highly keratogenous part of hair follicle. Now let's talk about very important point here, that stem cells are found in. They are found in Adamson's fringe. We have already talked about this. As you can see here, this is inverted V-shaped structure, that is Adamson's fringe, and in the Adamson's fringe there is the presence of stem cells. These red colored cells are stem cells. They are also found in basal layer of interfollicular epidermis, which means the basal layer of epidermis which is between two hair follicles. Let's see here, this is the epidermis and this is the basal layer of epidermis. In the basal layer of epidermis, this cell you can see in the red color is the stem cell. Stem cells are also found in base of sebaceous glands. This is the sebaceous gland. In the base of sebaceous gland there is the presence of stem cell. If you take a cross section of a hair follicle, what you can see is The inner part is known as medulla and the outer part is known as cortex. There is an index which is known as medullary index. Medullary index is the ratio of medulla and cortex of hair follicle. It is the ratio of medulla and cortex of hair follicle. There was a question which has been asked in DNB 2017 that medullary index in human hair is. The options were 1 is to 1, 1 is to 2, 1 is to 3, and 1 is to 4. And the correct option was 1 is to 3. Therefore, the medullary index in human hair is 1 is to 3. But in case of animals, the medullary index is 1 is to 2. This medullary index has an importance. What is that? This medullary index differentiates human hair from animal hair. Let's see some important layers of hair follicle here. This is medulla, this is cortex, cortex is followed by cuticle, cuticle is followed by Huxley layer, and Huxley layer is followed by Henle layer. Remember a very important point here that Henle layer, Huxley layer and cuticle are the layers of hair follicle. Now let's talk about types of hair follicles. There are three types of hair follicles. The first one is lanugo hair follicle, which is seen in fetus. The other two types are villus hair follicle and terminal hair follicle. Let's differentiate villus hair follicle from terminal hair follicle. Villus hair follicle is thin and terminal hair follicle is thick. Why villus hair follicle is thin? Because medulla is absent. And why terminal hair follicle is thick? Because medulla is present in terminal hair follicle. Villus hair is light in color because melanin is absent. And terminal hair follicle is dark in color because melanin is present. If we see the depth of these two hair follicles, the depth of villus hair follicle is till dermis and the depth of terminal hair follicle is till subcutis, that is hypodermis. It means the terminal hair follicle is more deep than villus hair follicle. Therefore, the terminal hair follicle is strong and the villus hair follicle is weak. Villus hair follicle is seen in prepubertal age groups and terminal hair follicle is seen in adults.